This is a presentation of the article Imaging Action Potential in Single Mammalian Neurons by Tracking the Accompanying Sub-Nanometer Mechanical Motion. What they actually did in this article was image the sub-nanometer mechanical motion associated with action potentials. A quick introduction to action potentials. An action potential is what transports the electric signal through neurons. It is initiated by a stimulus which causes channel opening, and this allows sodium ions to flow through a channel. This causes a membrane depolarization. After this, channels close and the membrane can re-establish its membrane potential. Article introduction. The main hypothesis of this article is that mechanical displacement is associated with action potentials. In this article, they present a novel technique to non-invasively measure the displacement associated with these action potentials. They did this by testing two experiments, one in human embryonic kidney cells and one in rat hippocampal neurons. Kidney cells cannot induce action potentials, but when applying a voltage to the cells, they still swell and shrink, and this can be measured by the technique. They measured 11 different rat hippocampal neurons and measured the displacement in the action potentials in these. An important method for this article is a patch clamp. In this experiment, patch clamp was used to induce electrical changes in cells. This is done in a so-called whole cell patch clamp setup. This is done by touching a neuron with a pipette and then inducing a Smyth suction, and this creates a hole in the membrane. This causes the inside of the pipette and the inside of the cell to be topologically identical. This was used in the two experiments described before, the HEK and the rat neurons. In the HEK cells, they varied the cell potential or forced the cell potential onto the cell, and then they measured the associated displacement with this. In rat neurons, they only initiated the action potential in neurons, and then they measured both the displacement and the voltage signal. Now the major question is, how can the displacement in neurons be measured so accurately? This is done by selecting a region inside and outside the membrane. In these regions, the light intensity is measured. Then a stimulus is applied and the intensities are measured again. If the intensity inside the cell increases and the intensity outside the cell decreases, the membrane shrinks, and this also goes the other way around. The values are also normalized to the total intensity to be able to compare them across the cells. The intensity difference is actually proportional to the displacement. Therefore, a calibration curve can be made by first choosing the two regions completely outside the cell, measuring the response, moving them slightly closer to the cell, and then measuring this response again. This is repeated until the two areas are completely inside the cell, and thus we get the calibration curve where we can take an intensity difference and relate it to a displacement. Now on to the results and discussion, first talking about the human embryonic kidney cells. It was found by the group that an average membrane displacement of 1.4 nanometers happened during the voltage stimulation. This agrees well with theory. But it was also found that the displacement differs a lot around the cell. So some places had a lot of displacement, while other areas almost saw no displacement. This is mainly attributed to protein, lipid, and cytoskeleton inhomogeneities. The group also performed fast Fourier transformation. This takes the measurement from the time domain to the frequency domain. This was mainly done to confirm the experiment's validity, and they found that they could actually get the frequency that they applied the voltage back from the results. Further result validation was done by measuring a point completely beside the cell where they saw no displacement, and also they measured the cell membrane but did not apply the voltage stimulation, and here they also saw no displacement. And now on to the results and discussion of the rat hippocampal neurons. It was found here that the maximal displacement is 0.2 to 0.4 nanometers at the maximal membrane potential, and this is also an average for the neuron. This compares to the predicted displacement of 0.1 nanometers. The displacement was again seen to be different across the cell, and thus a more detailed model is needed to describe the neurons. They investigated 11 neurons, and in all of these it was seen that the displacement and the potential match. That is, they show the same curve with the maximum displacement and the maximal potential being approximately at the same time. This is also the first instance of spatial information about action potentials. In conclusion, this group has shown that they can do non-invasive imaging of neuron sub-nanometer displacement that happens during action potentials. They have also shown that the mechanical displacement and the electric pulse of action potentials are synchronized. They have found a maximal displacement in neurons of approximately 0.2 to 0.4 nanometers. They see a large variability inside neurons, meaning that one place of the neuron can expand while another contracts. This seems to be a very time-consuming way to measure action potentials, but it do show some possibility of automatization if a program could be developed to analyze the images and calculate the edge displacement. With higher time resolution in these measurements, it could be possible to investigate action potential propagation through the axons of the neurons. The ultimate goal of this is to gain much more knowledge about action potentials. This could lead to knowledge about brain signaling and ultimately brain function. Another area that could have its knowledge increased by this research is the combating of neurodegenerative diseases, as described by Zimmerman and Tian.